Hello, everyone. I'm pleased to introduce Professor Bogdan Draganski. He's part of the organizing committee of Brain Fit for Life, which we started last year. And he's also director of the Center for Neuroimaging at CHUV. He's one of the pioneers in the Data Federation for integrated clinical phenotyping, data from imaging to behavioral data. And today he's going to talk about how to integrate neurobiological factors into the data registry and how to do a better predictive diagnosis of brain aging. So welcome, Bogdan. I'm really excited about your talk. Every talk so far has been fantastic. I'm sorry we had glitches, but that's part of the game.
Thank you very much, Lavinia. Now I'm sharing my screen. And after we heard this profound theoretical background from Parashkev and the big picture from Muriel, I'll come back again to the brain, and I'll be talking about large-scale brain studies in the aging population. Parashkev was advocating of having the big hospital data. I'll be doing exactly the opposite to bring a little bit of controversy for our discussion panel afterwards. I'll be much more counting the hairs in the eyelashes of my individuals rather than even looking at their intraocular distance. And my focus will be not on patients. It will be similar to Muriel's approach, focus on the general population. If we stare at the common knowledge that across the world, the population is aging, of course, it's not that much important for Europe. You see here on the left-hand side, the dark green squares already the numbers of people above 60, and the light green squares are the ones which will come additionally by 2050. You see Asia and Africa, much more the continents where this will proportionally rise. And if we focus then further on Switzerland, the situation now in yellow bars is that we have roughly across the different cantons 30 to 40 individuals above 65 for every 100 individuals that are younger, aged between 20 and 64. And the projection for 2045 is much gloomier. You see that this is rising there, particularly cantons where we are reaching even 70 age retired individuals on every 100 younger ones. That means we have even to overthink all the intergenerational contracts, asking ourselves who is going to take care of these individuals, besides, of course, the economic burden of this. When we focus then on the brain, and particularly my specialty is imaging of the brain, of course, we can pose a lot of interesting and unanswered questions to brain imaging, ranging from processes regarding to aging or neurodegeneration at the cellular molecular level, up to the macroscopic level, where we ask ourselves why, for example, in a neurodegenerative disorder like Alzheimer's disease, we have a particular spatial distribution of atrophy. And of course, asking ourselves, can we counteract this with some interventions? Can we exploit the brain plasticity, being by driving a cab across London or learning how to juggle in order to refresh, rejuvenate our brains? But the reality is actually different. Here I stole the idea from an old TED talk by Thomas Insel, and I projected it to one of the studies that I'll present in the general population that we actually started in 2013. But it covers the range between 2017 and 2027, where in 2017 we had 1,600 individuals aged 45 to 85 scanned with a brain MRI. And if we project a very approximative incidence levels of different diseases, we'll see that in the next 7 years, there will be a significant number that will have a depression in this time period, 300 out of this 1,600. 90 individuals will eventually develop Alzheimer's disease, 60 will get a stroke, 45 will get a Parkinson's disease. Well, at time zero, 2017, they did not have any of these disorders, at least overtly. That means the question is, how can we zoom in at earliest possible preclinical stage in order, again, to introduce interventions, what Jean-François Demonnet was talking about, what also Murray was advocating about, in order either to prevent or at least push back the disorders that are having such a impact on individuals' life and, of course, on family members. Murray was talking about this local longitudinal study that we are carrying out here in Lausanne. It started in 2003. It aimed at individuals aged 35 to 75 that were detected lege artis from the registry of the citizens of Lausanne. There were roughly 12,000 that fall into this category. They were contacted and roughly the half answered. That means we are talking about roughly 6,000 individuals that were recruited in 2003 and follow up every 5 years with the so-called somatic examination. That means they had some blood work looking for cholesterol, diabetes, chronic inflammation, etc. They were measured with all possible bioimpedance and weight and waist and hip, etc. metrics, including also some genetic information in the plain way of GWAS information. They had a very smart also assessment in terms of their mental health, rather than the person in white coat saying, well, I think that you are depressed. They used the so-called diagnostic inventory in genetic studies, which is a big questionnaire where you are asked about something and you say no, then you jump to the next question. If you say yes, then you come into something like a decision tree branching into different other questions. At the end of the day, we have a sparse matrix where you can run some algorithms, and in such a data-driven way run on, well, semi-objective data, you can establish either current or lifetime diagnosis, which is, I think, already a step forward. And 1,600 of these individuals also turned up for an MRI. And this is what I'll be talking about now in the next phase. That means already 400 from this 1,600 came here for a second brain scan. Of course, all of us are aware of bigger initiatives. Muriel was talking about the biobank, UK Biobank. You see also, we're talking about 1,600 individuals scanned, but the biobank has already 40,000. I mean, that means we are somewhere lightweight. We don't want to be, well, we want to be competitive, but we don't want to measure at that scale. I think what we can offer is something else. The interesting part here of this study, like also in the UK Biobank, are what kind of data are we acquiring besides these cardiovascular risk factors, besides this inventory of mental health? Well, there are a few interesting aspects. One of the big, I think, in-depth information wealth comes from the so-called HypnoLaus study, where roughly 2,000 individuals took part in polysomnography. That means they got all the cables around their head, looking at EEG rhythms during their sleep, measuring respiration, measuring also oxygenation of their blood. ActiLaus is another interesting also type of data. They were wearing 2 weeks long, 24 hours a day, track measurements that were looking at their physical activity, sleep, etc. Then we have also ophthalmologists looking at some eye problems, pain loss, looking at some metrics of pain. And to me, very interesting and a little bit more distant, geolouse and decibel-louse, looking at some georeferencing data related to also noise, either during the day or during the night around the area where the individuals are living. Well, what are we doing with our brain imaging? Of course, we are not radiologists. Our type of assessment is not putting the scans on a screen and thinking about some diagnosis. We're doing something that we call mesoscopic evaluation of brain anatomy. That means we're not just observing the whole brain from outside, like a pathologist will do before cutting it. We're not having, of course, the spatial resolution where we can look at the micron level resolution at some axons. We are trying to cover this in between. Of course, we are paying a certain price, and this is the level of granularity that in our domain is roughly a millimeter or a little bit less cube. And but the benefit, at least to conventional histopathology, is that we can do large-scale studies. Here in Lausanne, 1,600. In the UK, 40,000, going up to 100,000 soon. What do we do with these images? Well, we first of all have this expensive machine that we don't use for clinical purposes. You might say, well, what a waste. Well, not at all. Already for 20 years, there are solutions for assessing brain anatomy in a very elegant way, in a submillimetric precision, getting metrics either of the cortical thickness or of the volume across the whole brain. But the beauty actually links to what Parashkev is doing and was presenting. This is the fact that we are not just eyeballing the images and saying, well, this is good, this is rubbish, this is dementia, but we are using state-of-the-art statistical methods. That means all these blobs, colored blobs that you see on the right-hand side displayed on the surface of the brain is actually a statistical map of significance, let's say in the usual frequentist way, which is showing us some differences between groups or correlation with some parameters. Well, here we have a different approach, and this is where I was talking about counting the hairs on the eyelashes of individuals. We call it in vivo histology approach. This is something that I was lucky to co-develop with some colleagues in UCL some years ago. And here, instead of looking at thickness or volumes, we're trying to come closer to the neurobiological phenomenon behind. That means we're trying to measure tissue properties across the brain. What can we measure with MRI? We can measure myelin, either in the so-called white matter or intracortical myelin in the cortex. We can measure iron. We don't know if it's ferrous or ferric iron, but at least at the mesoscale, we can bring some interpretation of it. And then we're measuring also free tissue water. That means if we have edema, some cell loss with increase of extracellular space, then we're measuring this also directly. And of course, we are trying to do a reference to conventional histology. I'm not going to talk more about this, but on the right-hand side, you see also actually my brain with the distribution of intracortical myelin. It's a relative measure, and you see that in the sensorimotor areas, in the visual areas, and they're hidden also in the Heschel's gyrus, there is more myelin than elsewhere. This is something that we know already from more than 1 century ago, where Paul Flechsig was doing his drawings that you see just on the left-hand side of my brain. I think this is already promising. And why are we doing this? Well, a simple answer. There was one of the so-called milestone studies, you see it here on the left, that was published in 2004, that was trying to convince us that in the age range between 5 and 20 years, there is a loss of cortical thickness. That means everything what you see expanding across this, along this axis in blue color, we're losing actually brain cells. Well, this is biologically implausible, although it was so highly published, but it took 15 years that, again, in the very same journal, colleagues from Germany and the United States brought, using this technique that I presented, actually the empirical evidence that it's not actually a loss of cortex, it's much more the increase of intracortical myelin, which is plausible with, of course, brain maturation, that actually changes the contrast properties of the MRIs, that our smart algorithms were up to now interpreting, or we were interpreting, actually misled by our misunderstanding on contrast properties of MRI as loss of cortical thickness. But now when we can measure this, we can show, well, we're not losing cells, we're just gaining intracortical myelin. What we do now in this large-scale study is at that stage very simplistic. We're just trying, well, not to fish, but just to explore what kind of domains are having effects on brain anatomy. Well, here in this Manhattan plot, you see actually all the different domains that we are measuring, either cardiovascular risk factors in green color, inflammatory factors in yellow, lifestyle, like alcohol, tobacco use in gray, etc., etc. What is their correlation with all these different metrics in brain anatomy? And you see in a stringent GWAS type of statistical threshold, Bonferroni threshold here, the blue line, we still have here the cardiovascular risk factors with the major impact on brain anatomy. Well, nothing unexpected, but still it's convincing, I would say. And of course, we can browse in this way across the different metrics that we are having to get at least a little bit of orientation. Which domains do we have in our cohort with the major impact on brain anatomy? We can again zoom in, again in a very simplistic region of interest analysis way, and look then across these different subcortical regions, which are the different factors then one by one that affect the different tissue properties. I will just bring you here to this green squares in the second square that is representing actually the iron content of the brain. And we see a huge increase in iron content correlated with smoking. Well, this is interesting because actually the basal ganglia, the iron richest structure of the brain, they are of course abundant on dopamine receptors, and iron is needed actually for producing dopamine. And of course one can again link this to the role of basal ganglia in addiction, and this can, for example, be an interesting observation that will lead, of course, to more focused research. Then, of course, we can do our whole brain analysis and pose also some other questions, like which is the part of the brain which has the steepest decline with age, and what are the tissue properties contributing to this? Of course, age, as also Parashkev said, has profound impact on the brain. If you look at the upper row, the whole brain is changing, decreasing volume, decreasing myelin, getting less iron and more actually free tissue water. But if we then ask specifically, well, among all this age-related atrophy and changes in tissue properties, which is the region that has the steepest decline, we see it's the primary motor cortex. I was pretty surprised. I thought it would be much more areas linked to memory or attention, but it's the motor cortex. And you see it very convincingly here. It's the volume decrease that is paralleled by loss of intracortical myelin, loss of intracortical iron, because iron is also needed to by the oligodendrocytes to continue the process of remyelination, and also in parallel, again, absolutely in the very same region, increase in tissue water. Now it makes sense. It's not just saying, well, we are losing volume there, but indeed we are trying to get in this mechanistic explanation of tissue property changes driving this atrophy. We're getting also, of course, some other metrics that are interesting here. The results, again, of a very simplistic study that was seductive because we had so many individuals, where we're looking at some polysomnography parameters and asking ourselves, well, is it the degraded sleep in sleep apnea, or let's say lega artis, the description, sleep disordered breathing, that has an impact on brain anatomy, or is it something else? And across all the parameters that you can imagine coming out of a polysomnographic examination, it's only actually the oxygen saturation that is, of course, the main feature of sleep apnea that has a negative effect on these brain regions, particularly the hippocampus, the basal ganglia, thalamus, and some cortical regions. That was interesting, and of course, this merits further study. Well, besides this type of assessment of tissue properties, we have another technique which is sensitive to diffusion of tissue water. And here we're using some more sophisticated biophysical models. Of course, I was talking about the price that we are paying, that we are not that much zooming in terms of spatial resolution because we don't have the ability doing this with the current machines, but at least we can have biophysical models that we can, of course, then we can fit our data to this and make also inferences on certain features that are at the microscopic levels. And here, with this diffusion-based imaging, we are getting some information about the density of axons within each pixel or voxel. And if you remember, I was presenting also the other technique, which is delivering us also metrics indicative for myelin content. If we marry these two types of information, axonal density and quantity of myelin, we can also estimate across the whole brain the whole so-called G ratio, the ratio between the diameter of an axon and the myelin sheath around, that you see here on the right-hand side is accessible only to electron microscopy. Interesting. Well, with these metrics on tissue properties and on G ratio, we can, of course, look in this population on lifetime trajectories, something what also Muriel was advocating. Of course, in a lega artis way, we can look at the best model fit. Is it a linear decline between age 20 and 80, or is it more an inverted U-shape, as you see? And indeed, this is the case. But these are representations of different metrics and across different fiber tracts in the brain. And you see more or less the majority of them are following this inverted U-shape or true U-shape. This is a way how we can also get a little bit more information about the white matter of the brain that was up to now not that accessible to MRI. And particularly, this is very interesting when we're talking about microvascular lesion and their impact on cognition, what also Jean-François was alluding to. Well, we can take also the big picture. We can ask ourselves, well, if we have such rich information about the different tracts, can we, in a data-driven way, ask the question if there are different ways how white matter is aging? And actually, we see 5 different modes or ways how white matter is aging. I'm not going to go through all of this. You see them here on the right-hand side. This is a fully data-driven approach. And it's interesting because somehow it gives the impression that it's much more linked to the different systems, be it the limbic system, be it the frontal projection fibers, etc. And it's only one mode where we have actually interhemispheric connections. This makes sense. This is interesting because this unanswered question about distribution of neurodegeneration may be answered here by differential vulnerability of tissue properties across the white matter. Well, the last approach that we are using is very simplistic covariance approach, where we're looking how the different parts of the brain covary in terms of brain tissue properties. And if we look actually what is the effect of aging on this covariation, which is interpreted as a metric of homotopy or metric of structural connectivity, we see actually in the different parts of the brain that somehow aging is causing a uniformity across the cortical and subcortical areas. That means something what in young age was much more differentiated, with increasing age is getting much more uniform. Interesting observation. Of course, this also merits further exploration. Well, the last thing, it's much more like a teaser for further discussion, is this work that we're doing with Stefan Joos from here, the Polytechnic in Lausanne, who is a specialist in georeferencing. And here we are trying to build the map of Lausanne on a brain map and vice versa. That means here we are using state-of-the-art methods, which are, at least surprisingly to me, very similar between the way how we look at brain topography and how Stefan is looking at the topography of Lausanne. And here we are looking at some clusters which are represented here in 2 extreme colors, either red or blue. All the white dots are the participants of our Colau study, the cohort in Lausanne. And here we are detecting some clusters, either with low MMSE, this overall score for brain cognition, or with high MMSE. This is corrected, of course, for socioeconomic status, because you can say, well, this is, of course, a big confounder. But we see a certain distribution of this either low or high cognitive performance across the ages that we're looking at in the city of Lausanne. And of course, we are looking what are the factors and what are the implications of proximity to green areas or the high or low nocturnal noise to these metrics. Highly interesting. I'm very fond of this type of research. Well, I'll do my wrap up because I'm at the end of my presentation. I think that counting the hairs on the eyelashes, that means this novel quantitative structural MRIs, has a potential becoming a game changer. Not in clinics. Against this is exactly against what Parashkev was propagating for the hospital or patient-related work here, looking here at some preclinical stages. I think the quantitative character of this imaging has particular importance for longitudinal studies, because numbers should not change if nothing is changing in the brain, which is a important point. We can study indeed meso and micro scale features of brain anatomy non-invasively without opening the skull. And this is helping us to understand what we are always looking after, the relationship between brain structure and function. Challenges, of course, data quality. We need more validation with histology. And again, to wrap up, I think we need a more proper generative model that is integrating all this information. Thank you very much for your attention.
Thank you, Bogdan. It's been really a great insight into how monitoring structural changes with aging. And I think we will switch to the panel discussion where all speakers are present. And we also have polls for the audience. So it will be an interactive session. Thank you.